বিসমিল্লাহিররহমানিররহিম। আমাদের আজকের আলোচ্য বিষয় হলো hypersensitivity, introduction to hypersensitivity and details of type 1 hypersensitivity. Bismillahirrahmanirrahim. Rabbi zidni ilma, Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatan min lisani yafqahu qawli. আমাদের আজকের hypersensitivity relation planটা হলো what is the definition of hypersensitivity. There are two types of definition, classical and working definition, a little bit about historical background, importance of understanding hypersensitivity. Orthat, why we need to understand the hypersensitivity, because most of the many diseases, their pathophysiology depends on understanding hypersensitivity, autoimmune as well as microbial disease and even many diseases. Classification of hypersensitivity and details of type 1 hypersensitivity, mediators, stages, treatment, and desensitization, very important thing. What is the working definition of hypersensitivity? It's very simple. Adverse clinical reaction to the antigen, or when the immune system does something bad to the host, that is tissue damage, that is hypersensitivity. Previously, it was thought that immunity, that is immune response, is always beneficial to the host. But subsequently, it is found that we all know that excess of anything is bad. Similarly, it applies to the hypersensitivity, that when immune response reacts excessively, then it is not good for our health. So that is, it causes some damage to the host, and that time it is known as hypersensitivity. Why we are calling it working definition? Because there is a classical definition, and many of our students, they memorize this definition. Classical definition is inappropriate or excessive immune response in a pre-sensitized host leading to tissue damage. এবং আমাদের অনেক টিচাররা এই শব্দগুলো না বললে এখনো বলে যে হয় না ডেফিনিশনটা। কয়েকটা শব্দ, একটা হলো ইনঅ্যাপ্রোপ্রিয়েট অর এক্সেসিভ ইমিউন রেসপন্স, কাকে? প্রি-সেন্সিটাইজড হোস্ট দ্যাট লিডস টু টিস্যু ড্যামেজ। কিন্তু এখন দেখা যাচ্ছে যে অনেক হোস্ট যদি ইনেট ইমিউনিটিতে, সেখানে তো প্রি-সেন্সিটাইজড হয় না, সেখানেও হতে পারে। সেজন্য রিসেন্ট ডেফিনিশন রয়ের থেকে চেঞ্জ করা হয়েছে। Hypersensitivity can also arise from direct interaction of the inciting agent with the elements of the innate immune system without intervention by acquired immune responses. তো সব মিলে আমাদের ওয়ার্কিং ডেফিনিশন হলো when the immune system does something bad to the host, that is hypersensitivity. পরিষ্কার? Now let us look into the historical background of hypersensitivity. Before 1900, it was thought that immunity is always good. But in 1902, two scientists, Charles Robert Richet and Paul Portier, they did an experiment. They injected toxin into dogs. And when second dose given after 1 week, instead of protection, dogs become ill. Excessive salivation, defecation, difficulty of respiration rate within minutes. So this is an exception to prophylaxis. Prophylaxis means protection. So they named this phenomenon as anaphylaxis. That means away from prophylaxis. It is away from prophylaxis. And this is the first example that immune system can cause harm. And this is the Charles Robert Richet. They won the Nobel Prize in 1930. Let us discuss about the classification of hypersensitivity. They can be classified on different bases. First of all, on the basis of onset of action, after introduction of the antigen, how fast it occurs. There are basically two types we will discuss, immediate and delayed. This can also be classified on the basis of mechanism of action. We know immune system acts on two types, cell-mediated and antibody-mediated. So here also, which component is involved predominantly? And based on the two things, this onset of action, mechanism of action, Coombs and Gell, they give a combined classification, type 1, 2, 3, 4, like this. So based on onset of action, it can be immediate hypersensitivity and delayed hypersensitivity. Immediate means within seconds or minutes, and delayed means within hour to days even. Based on mechanism of action, as I have said, it can be antibody mediated and it can be cell mediated. So when the antibodies are involved, they are called antibody mediated, type 1, 2, 3, and 5. And only 4 is cell mediated. So Coombs and Gell in the early 1970s, they proposed a classification method for hypersensitivity, type 1 to 5. Type 1 is anaphylactic hypersensitivity. As the name suggests, there is anaphylaxis, a rapid some reaction. Type 2, also known as cytotoxic hypersensitivity. Type 3, immune complex hypersensitivity. Type 4 is delayed or cell-mediated hypersensitivity. And type 5, not named by Coombs and Gell, that are added, it is stimulatory hypersensitivity. If you focus on the names, the name suggests many things. Say, for example, cytotoxic hypersensitivity. That means in this hypersensitivity, there is cytotoxicity of the involved cell. Immune complex hypersensitivity, that means immune complexes are formed, and on deposition of immune complexes, the pathogenesis occurs. And delayed or cell-mediated, delayed means it occurs after many hours, there is even after days, and cell-mediated component is involved, so this is. And stimulatory hypersensitivity, instead of direct damage to the cell, there is stimulation of the cell, so it is known as stimulatory hypersensitivity. So you see, this is the Coombs and Gell classification, diagrammatic. Say in type 1 hypersensitivity, as we have said, antibodies are involved, IgE antibody. This antibody is soluble, and the cell involves are mostly mast cell, and there is the diseases. Examples are allergic rhinitis, asthma, systemic anaphylaxis, etc. We will discuss in details in this lecture. Type 2 is IgG antibody mediated, and here complements are involved and results in cytotoxicity. Type 3 also IgG involved, complements are also involved, but immune complexes are formed, and this pathogenesis depends on the deposition of immune complex in different sites. Type 4 is usually cell-associated or macrophage or soluble antigen. Macrophage and cytotoxic T cells are involved, and that is different types of hypersensitivity. Let us discuss in details about the type 1 hypersensitivity. Type 1 hypersensitivity, also known as immediate hypersensitivity, allergy, anaphylaxis, or IgE-associated immune response. So, this is, if you remember these names, it is clear. And this figure shows how it acts, but we will go in details by other figures. Now, let us explain the terms. What is allergy and allergy, from where the term came. What is allergy? Allergy means allergy, a biological hypersensitivity in certain individuals to substances which in similar amounts and circumstances do not have an effect on others. আমরা একটা এক্সাম্পল দিতে পারি যে গরুর গোশত খেলে সবার একরকম হয় না, চিংড়ি মাছ খেলে সবার একরকম হয় না। তো যাদের হয় এই চিংড়ি মাছ বা গরুর গোশত বা ফুলের রেণুতে গেলে ওই যে অল্টারনেট যে অ্যালার্জিক যে অল্টার্ড ইমিউন রেসপন্সটা, দিস ইজ অ্যালার্জি। In other words, allergy is an unusual or exaggerated response to certain substances, but not to all people, in certain people. So what is allergen? A substance capable of producing an allergic reaction is known as allergen. It is just like antigen. It can be anything. And Hippocrates in 462 BC, he said, one person's food may be in another person's poison. যেরকম আমরা দেখি চিংড়ি মাছ একজন মজা করে খাচ্ছে, এই চিংড়ি মাছ খেলে আরেকজনের গায়ে অনেক রকম চুলকানি শুরু হচ্ছে। তো এটাই হলো অ্যালার্জি। তো কাদের এটা হয়? This allergy termটা কোত্থেকে আসলো? This term allergy was coined in 1906 by von Parquet. উনি একজন অস্ট্রিয়ান ফিজিশিয়ান ছিলেন এবং উনি এই টার্মটা করেছিলেন। There are two types of allergies, active allergy and hidden allergy. Active allergy causes very visible, uncomfortable allergic reaction including rash, swelling, wheezing, runny nose. সবাই এটা বুঝতে পারে যে আমার অ্যালার্জি হয়েছে, লোকজন বলে। হিডেন অ্যালার্জি, এই টার্মটা আপনারা আগে শুনেন নাই। They cause few visible allergic reaction. Patients complain of poor function of the body or mind, including fatigue, anxiety, mental fogginess, back pain, arthritis, and অনেক সময় তারা বলে যে এটা আমার কেমন কেমন যেন লাগে। এই কেমন কেমন জায়গায় যখন কোনো কারণ খুঁজে পাওয়া যায় না, অনেকেই তাদেরকে মানসিক রোগী বলে আখ্যায়িত করে, বাট এটাও একটা হিডেন অ্যালার্জি। এই জিনিসটা আমাদের একটু মনে রাখলে ভালো হয়। এবং অনেক পেশেন্ট, মোস্ট অ্যালার্জি সাফার্স ফ্রম বোথ টাইপস। যখন এই রানিং রোজ এগুলো হয় তখন তার ওই মেন্টাল ফ্যাটিক, ফগিনেস, ব্যাক পেইন, আর্থারাইটিস, পুয়ার ফাংশন অফ দ্য বডি, এগুলো হতে পারে। এই দুটো মিলেই সাধারণত ম্যানিফেস্টেশন হয়। কখনো কখনো হিডেন অ্যালার্জিটাই প্রিডমিনেন্ট হয়। সো হিডেন অ্যালার্জি, এই টার্মটা আমরা নতুন করে মাথায় রাখলাম। What are the allergens? Allergens are antigens that induce production of a specific immunoglobulin E antibody, এবং তারা অন্য কোনো অ্যান্টিবডি তৈরি করে। এদের এক্সাম্পল কি? এরা প্রোটিন হতে পারে। They can be protein, polypeptide, polysaccharide, nucleic acid, low molecular weight chemicals, etc. এদের ক্যারেক্টারিস্টিক্স কি অ্যালার্জেন? Usually they are low molecular weight, less than 40 kilodalton, water soluble and stable. Route, they can enter, we all know, they can enter through skin, respiratory tract, GI tract, and eye. সবাই আমাদের কারো না কারো এক্সপিরিয়েন্স আছে, চোখ জ্বালা পোড়া করে। GI tract দিয়ে ঢুকলে আমাদের ডায়রিয়া হতে পারে। রেসপিরেটরি ট্র্যাক্ট আমরা সবাই বুঝি, হাঁচি কাশি, স্কিন চুলকায়। Common allergens are house dust, mite, microorganism, animal protein, even some antibiotic, insect poison. কারো কারো মশা কামড়ালে বা সাপ পোকা কামড়ালে অনেক বেশি ফুলে যায়, এগুলো অ্যালার্জেনের এক্সাম্পল। So common allergens associated with type 1 hypersensitivity, we can classify protein, foreign serum, vaccine, plant pollen, drugs, ফুড, নাট, এত সুন্দর একটা খাবার নাট, এটাও কারো কারো অ্যালার্জি হতে পারে। সি ফুড, এগ, কেউ কেউ হাঁসের ডিম খেতে পারেন না। ইভেন মিল্কেও কারো কারো অ্যালার্জিক আছে। ইনসেক্ট প্রোডাক্ট যেমন বি, অ্যান্টিভেনম, ডাস্ট মাইট, এটসেট্রা। মোল্ড স্পোর, অ্যানিমাল হেয়ার অ্যান্ড ড্যান্ডার, ল্যাটেক্স, এটসেট্রা। So another term is atopy and atopic individuals. Atopy describes that the atopic persons, they produce excess amount of immunoglobulin E in response to certain allergen, and they suffer mostly from type 1 hypersensitivity, asthma, eczema, hay fever, urticaria, etc. These individuals normally have a strong familial history, autosomal transmission of. So atopic individuals are a group of individuals who produces more immunoglobulin E in response to certain antigens, and they suffer from asthma, eczema. Actually, 10 to 15% of our population, they are atopic to certain environmental allergens. HLA versus allergen responsiveness. Some allergens response have a relationship to HLA, HLA-DR2 and HLA-A2, they are high responder to low dose of certain allergen, like reagware. HLA-B28, they are also high responder to reagware and also associated to other forms of hyperimmunity, autoimmunity. So HLA have got some relationship. Risk of allergy. If both parents are atopic, then the sibling, their children, they have got a risk of 50%. If one parent is involved, 30%. If none of the parents are atopic, even then they have got a chance of 15% being atopic individuals. IgE is the antibody, only antibody that involves in type 1 hypersensitivity. We all know that IgE concentration in normal individuals are very low, and usually after production they remain attached to the mast cell with their Fc fragment. Interleukin-4, they promote B cells to differentiate Ig to produce IgE. They differentiate the B cells to become IgE producing B cell. What are the phases of type 1 hypersensitivity? There are 3 phases: sensitization phase, effective phase, sensitization phase, triggering phase, and effective phase. Simply we can, sensitization means the atopic individuals, when they comes in contact with some allergen, say food allergen or inherent allergen, this allergen will be taken up by the dendritic cell, antigen presenting cell. They will go to the B cell, and this B cell will produce excessive amount of immunoglobulin E. This immunoglobulin E will immediately after production, they go and bind to the mast cell, but there will be no clinical sign symptom. Triggering phase, the same person, when again they are re-exposed to the same allergen, again this allergen is taken up by the macrophage, and it is again producing this immunoglobulin E. This immunoglobulin, this allergen, they go to the immunoglobulin E that is bind on the mast cell, prefixed, and there will be some degranulation of the mast cell, resulting in many chemical mediators we will discuss in detail. And these chemical mediators, ultimately there will be some end organ responses like vascular leak, smooth muscle contraction, mucus secretion, and all this will lead to the sign symptoms either local or systemic. So this is the triggering phase. If the sensitized person is not exposed to the antigen again, there will be no sign symptom. So you see, when the antigen for the first time exposed to atopic individuals, it will produce the immunoglobulin E. This immunoglobulin E will bind to the mast cell by their Fc fragment. Their FAB fragment will remain free. When the same antigen again enters in the body, they cross-link with the FAB fragment. What is cross-linking and linking? এটা হলো বাংলা এক্সাম্পল দেওয়া যায় মানব বন্ধন। যদি আপনি আপনার দুটো হাত দিয়ে আরেকজনের দুটো হাত ধরে থাকেন, সেটা হলো লিঙ্কিং। যদি আপনার একটা হাত একজনকে দেন, আরেকটা হাত আরেকজনকে দেন, আমরা যেরকম মানব বন্ধন করি, সেটা হলো ক্রস লিঙ্কিং। এই ক্রস লিঙ্কিং এর ফলেই ডিগ্রানুলেশন হয়, লিঙ্কিং এর ফলে হয় না, মনে রাখতে হবে। এবং তখন আবার মেডিয়েটর গুলো রিলিজ হবে, রিলিজ হয়ে ক্লিনিক্যাল এফেক্ট হবে। সো আমরা এই অ্যানিমেশনটা দেখি যে কিভাবে ইমিউনোগ্লোবুলিন ই তৈরি হচ্ছে। আমরা খেয়াল করেন যে প্রথমে একটা অ্যান্টিজেন আসলো, অ্যান্টিজেন এসে একটা বি সেলকে স্টিমুলেট করলো। আপনারা খেয়াল করেন সারফেসে অ্যান্টিজেন এসে এখানে বি সেলকে সে স্টিমুলেট করলো। বি সেল স্টিমুলেশন হয়ে অ্যাক্টিভেশন হয়ে লার্জ অ্যামাউন্ট অফ ইমিউনোগ্লোবুলিন ই তৈরি করবে। সেই ইমিউনোগ্লোবুলিন ই গুলো কোথায় যাবে? ইমিউনোগ্লোবুলিন ই যে মাস্ট সেলের গায়ে যে এই লেগে থাকে। এখন যদি আবার একটা অ্যালার্জেন আসে, দেয়ার উইল ক্রস লিঙ্কিং, এন্ড ক্রস লিঙ্কিং হলে এই মাস্ট সেল ডিগ্রানুলেশন হয়। and degranulation will lead to the clinical signs symptoms. Very interesting, just to see. So, এই অ্যানিমেশনটাকে আমরা এখানে আবার নিয়ে এসেছি, আমরা জাস্ট এটাকে, it is very important. You see the antigen, this is the triggering phase. Formed immunoglobulin E is attached in the mast cell surface by their FC portion on the FC receptor. When the antigen again enters during the triggering phase, they cross-linked the immunoglobulin E. Following the cross-linking, there is some signal, and this signal leads to the degranulation of the mast cell. This degranulation is a physiological process. The mast cell will not burst, just it will degranulate. This degranulate, the granule contains the histamine, protease, chemotactic factors like eosinophil chemotactic factor, neutrophil chemotactic factor. These are known as primary mediators. And some cytokines will also be released. And there is another mechanism, membrane, this signals for activation of phospholipase A2. There are three signals. Signals for degranulation, signals for cytokine gene activation, and signals for activation of phospholipase A pathway. And that pathway will lead to arachidonic acid and later activating factor, that is leukotriene B4, C4, D4, and prostaglandin D2. All these are secondary mediators. So activation of mast cell in type 1 hypersensitivity and release of mediator, that is eosinophil chemotactic factor, neutrophil chemotactic factor, all this you will see. Now this slide shows, other than cross-linking, what are the other mechanisms by which a mast cell can degranulate? You see, this is the allergen cross-linkage of cell-bound IgE. This is one mechanism. There is another mechanism is, say, if anti-isotype antibody, there is antibody which is formed against the FC portion, that can also lead to mast cell degranulation. And anti-idiotype antibody, say, instead of antigen, an antibody is formed against the FAB fragment. Here antibody is formed against FC fragment. Here antibody is formed against FAB. This is FC, this is FC. So this is anti-isotype antibody, anti-idiotype antibody. That can also lead to degranulation of the mast cell. C, cross-linkage by chemical. Say any chemical, if can cross-link this immunoglobulin E bound to the mast cell, that can also lead to mast cell degranulation. Cross-linkage of IgE receptor by anti-receptor antibody. Say these receptors, FC receptors, are not occupied by immunoglobulin E, but some antibody against the receptor bind here, cross-link here, that can also lead to mast cell degranulation. Finally, enhanced calcium influx by ionophore that increases membrane permeability to calcium, that can cause mast cell degranulation. So there are total 5 mechanisms. Besides the type 1 hypersensitivity, these other mechanisms, they are not called as anaphylaxis hypersensitivity, they are called anaphylatoxins. These are mechanisms. So the same picture in a different way. Number 1 is by cross-linking by antigen. Number 2 by anti-idiotype antibody. Number anti-isotype antibody. This is anti-idiotype antibody. This is anti-FC receptor antibody. And by lectin and chemicals also, we have said. What are the products of human mast cells? So you see, there are some granules, there are some cytokines, chemokines, and leukotrienes, C4, D4, and they have got different type of activities, including epithelial damage, mucus hypersecretion, airway narrowing, pro-inflammatory cell attraction, and these cytokines, they have got activation of eosinophil and all these things. So time of chemical mediator release. They are released in two phases, early phase and late phase. Early phase means 2 to 3 minutes to 6 hours. Early phase, histamine, eosinophil chemoattractive factor, and neutrophil chemoattractive factor are released within 2 to 3 minutes to 6 hours. Late phase, more than 6 hours, within 24 hours, leukotrienes and platelet activating factor, vasoactive amines. So, histamine, prostaglandin, leukotriene, C4, D4, all are known as spasmogen because they constrict bronchial smooth muscle, the mucosal edema, mucus secretion resulting in some asthma-like symptom. Let us some disease. We can give some examples that are type 1 hypersensitivity mediated disease. Bronchial asthma is the common example. Allergic rhinitis, urticaria, anaphylactic shock, food allergy, eczema, and hypersensitivity. এখানে একটা জিনিস মনে রাখা দরকার যে হাইপারসেনসিটিভিটি মানেই হলো বিভিন্ন রকম ডিজিজ, আর ইমিউনিটি সেটা হলো প্রোটেকশন। সো আমাদের হাইপারসেনসিটিভিটি গুলো বুঝতে হবে ডিজিজ বোঝার জন্য। সো এবং তাহলে তার ট্রিটমেন্টও সোজা, আপনি যদি ডিজিজ প্রসেসটা বোঝেন। সো all these are type 1 hypersensitivity, bronchial asthma, allergic rhinitis, urticaria, food allergy, all these things. And you see the IgE-mediated allergic reactions. This is a repetition of the systemic anaphylaxis. Usually, it is caused by common allergens like drug, serum, venom, and root is intravenous edema. Wheel and flower, all insect bite, allergy testing subcutaneous, allergic rhinitis, inhalation, bronchial asthma, inhalation, food allergy following by oral ingestion, these food products, and their manifestation is also different. Food allergy is manifested by vomiting, diarrhea, etc. Bronchial asthma manifested by bronchial constriction, allergic rhinitis by edema of nasal mucosa, wheel and flower, local vasodilatation, and systemic anaphylaxis by systemic manifestation. So, primary and secondary mediators, histamine, serotonin, eosinophil chemoattractive factor, neutrophil chemoattractive factor, protease, all these are primary mediators. Platelet activating factor, leukotrienes, prostaglandin, bradykinin, cytokines, interleukin 1, interleukin 4, interleukin 13, interleukin 3, 5, many cytokines are involved. And you see their responses are also from local to systemic, so many manifestations. Mostly, they increases vascular permeability, smooth muscle contraction, eosinophil chemotaxis, their name also suggests, platelet aggregation, degranulation, and all these are responsible for the clinical feature of type 1 hypersensitivity mediated disease. Let us discuss about the laboratory testing. There are many types of tests, specific, non-specific, of which important tests are skin test, IgE level, eosinophil count, and skin test. It is there are panel of allergens, and you take a very small amount of the allergen and give on the forearm skin, and you look for the edema. And by this test, you can detect which allergen is involved in your type 1 hypersensitivity. This is available in Bangladesh in some places. Say here, you can see grass allergen is responsible for type 1 hypersensitivity. We all know eosinophilia occurs in type 1 hypersensitivity, and eosinophilia actually is a protective mechanism. It protects us. This eosinophilia, they have the mediators, and one of the important mediators is histaminase, so it helps us. The mediators of eosinophils are major basic protein eosinophil cationic protein, eosinophil denatinic peroxidase, leukotrienes, interleukin alpha, many leukotrienes are involved here. So types of receptors for immunoglobulin E, there are two types of receptors, high affinity receptor and low affinity receptor. High affinity receptors are expressed on mast cell and basophils. Low affinity receptors are expressed on lymphocytes. And these are the sign symptoms, graphical presentation of the sign symptoms in type 1 hypersensitivity. Systemic, you see there is itching, conjunctivitis, rhinitis, angioedema, hypotension, there is laryngeal edema, urticaria, bronchospasm, dysrhythmia, GIT, some symptom angioedema, and all these things. So let us discuss about the treatment and prevention. So treatment is based on, first of all, you can avoid the allergen, that is environmental control. You can remove the IgE by plasmapheresis, or immunological treatments is desensitization. Desensitization, and there are some chemical treatment also, inhibition of mediators release, and histamine antagonists like antihistamine, and late phase inhibitors like steroid, this and all the other chemical treatment. But we are, and you see, antihistamine, cromolyn, sodium, theophylline, epinephrine, cortisone, these are chemical treatment of the type 1 hypersensitivity. But we are interested on the immunological intervention. And immunological intervention is very interesting, it is known as desensitization. There are two types of desensitization, one is acute and another one is chronic. In acute desensitization, very small amounts of antigen at 15-minute intervals to form antigen antibody, this IgE complex on a small scale, which will not degranulate mast cell. It is usually used before during the ADS or ATS therapy. Chronic desensitization is small doses of allergens are injected at weekly intervals where immunoglobulin G antibody is produced, which blocks the antigen by binding with it and does not allow the antigen to reach to the immunoglobulin E bound to the mast cell. অর্থাৎ এখানে ইমিউনোগ্লোবুলিন ইর পরিবর্তে জি তৈরি হয়, জিটা সেই অ্যান্টিজেনকে বাইন্ড করবে, সেটাকে ইমিউনোগ্লোবুলিন ই পর্যন্ত পৌঁছে দেবে। এবং severity may be reduced by desensitization shot, that is allergic shot. It is thought that when very dilute allergen is given by injection, it stimulates the production of immunoglobulin G and A, not E. This immunoglobulin G and A then act as blocking antibody to bind and neutralize much of the allergen in the secretions before it can bind to the mast cell bound immunoglobulin E. These shots also appear to suppress production of immunoglobulin E by inducing tolerance or by activating suppressing T suppressor cell. দুটো জিনিস, অর্থাৎ আমি অল্প পরিমাণে ডাইলুট অ্যামাউন্টে যদি দেই, ই তৈরি না হয়, অ্যালার্জিক পারসন, অ্যাটোপিক ইন্ডিভিজুয়াল গুলো ই তৈরি না করে ইমিউনোগ্লোবুলিন জি এবং এ অ্যান্টিবডি তৈরি হবে, যারা ওকে নিউট্রালাইজ করে ফেলে এবং ওই মাস্ট সেল পর্যন্ত পৌঁছতে দেয় না। আর ইমিউনোগ্লোবুলিন ই আরেকটা মেকানিজমে সাপ্রেস হতে পারে বাই সাপ্রেসিং by activating T suppressor cell. এছাড়া এখন আরেকটা there is a new experimental approach. If we can produce the monoclonal antibody that have been made against the FC receptor, FC portion of immunoglobulin G, this blocks the attachment of the immunoglobulin E to FC receptor on the mast cell and basophil and subsequent তৈরি হবে, কিন্তু এরা ওই FC receptor এ বাইন্ড করতে পারবে না। এটা একটা আমরা ছবি দেখাই, এবং যদি বাইন্ড করতে না পারে তাহলে anti-IgE binds to the IgE producing B cell, they cause apoptosis. আমি ছবিটা আমরা দেখাই যে সাধারণত যদি এই FC রিসেপ্টর এগেইনস্টে আমি একটা অ্যান্টিবডি তৈরি করি, মনোক্লোনাল হিউম্যান অ্যান্টিবডি, ওই অ্যান্টিবডি গুলো এখানে বান্ডে তাকে বাইন্ড করতে দেবে না। বা আর যদি আমি IgE এর FC পোর্শনের এগেইনস্টে অ্যান্টিবডি তৈরি করি, তাহলে সেই মনোক্লোনাল অ্যান্টিবডি গুলো IgE তৈরি হওয়ার সঙ্গে সঙ্গে তার সঙ্গে বাইন্ড করে যাবে। তো সেই IgE আর তখন FC রিসেপ্টরে আসতে পারবে না। তাহলে আমি এই রিসেপ্টরের এগেইনস্টেও অ্যান্টিবডি তৈরি করতে পারি, কিংবা IgE FC পোর্শনের এগেইনস্টেও করতে পারি। তখন আর মাস্ট সেল রিজেনারেশন হবে না, আর মাস্ট সেল রিজেনারেশন না হলেই তখন আমার টাইপ 1 হাইপারসেনসিটিভ হবে না। এটা বোঝা গেল আশা করি। অর্থাৎ এই রিসেপ্টরের এগেইনস্টেও অ্যান্টিবডি তৈরি করতে পারি, অথবা IgE এর FC পোর্শনের এগেইনস্টে অ্যান্টিবডি তৈরি করতে পারি। সো থ্যাংক ইউ ভেরি মাচ। আশা করি এটা বোঝা গেছে। আসসালামু আলাইকুম ওয়া রাহমাতুল্লাহ।